In this next video, we're going to explore how we can separate mixtures of amino acids using different chromatography techniques. In the previous video, we developed an appreciation for how amino acid structures and charges can change based on the pH of the solution they're in. So we're now going to talk about how different techniques can be used to separate amino acids that are in a mixture. We can exploit two characteristics of amino acids in our efforts to separate them. We can exploit two characteristics of amino acids in our efforts to separate them from each other: differences in charge and differences in polarity of their R groups. As we saw in the previous lecture, charge is affected by pH. Generally speaking, increasing pH tends to deprotonate functional groups. This tends to make the net charge on an amino acid more negative. Decreasing pH tends to protonate amino acid functional groups, making the net charge more positive. Polarity of the R group influences solubility in a given solvent. Polar amino acids dissolve in polar solvents, while nonpolar amino acids dissolve in nonpolar solvents. Chromatography refers to a set of methods and techniques that can be used to separate mixtures into their component parts. In the laboratory, we're going to be focusing on separating mixtures of amino acids. We'll be using two chromatography methods: ion exchange chromatography and thin layered chromatography. Thin layer chromatography separates amino acids based on their differences in polarity. The steps are fairly straightforward. First, you spot samples onto the TLC plate, which is called the stationary phase. Be sure to mark the line where the spots are made lightly with a pencil. Place the TLC plate into the TLC chamber that has a small volume of the appropriate solvent in the bottom. The chamber should be covered to prevent evaporation. The solvent will migrate up the TLC plate by capillary action. If the solvent is polar, then polar amino acids will move more readily by dissolving into that solvent, and they're going to be carried with the migrating solvent up the TLC plate. Nonpolar amino acids will tend not to dissolve into the polar solvent, and therefore shouldn't migrate as far as the polar amino acids. Thus, the amino acids in the mixture can be separated based on their polarity. Now, it's entirely possible that you would not be able to visualize the results of this experiment because amino acids are colorless. So you may have to actually develop your TLC plate by spraying ninhydrin on the TLC plate, which will interact with amino acids to produce a purple color. So TLC is going to separate amino acids based on their differences in polarity. Once the solvent reaches about a ½ centimeter from the top of the TLC plate, remove the TLC plate from the chamber and make a thin line where the solvent front is. The TLC plate is then sprayed with an ninhydrin, which allows for the visualization of colorless amino acids as purple spots. The spots are marked on the TLC plate, usually by indicating the center of the spot. RF values can then be calculated using the formula of RF equals the distance traveled by the sample divided by the distance traveled by the solvent. Whereas thin layer chromatography takes advantage of amino acid polarity, ion exchange chromatography takes advantage of the amino acid net charge. In ion exchange chromatography, a column is packed with extremely fine beads made of a resin. Each bead carries a charge, either a negative charge or a positive charge. Amino acids will stick to the beads and consequently the column for varying amounts of time depending upon their charges. In the middle panel, we're showing a resin bead that carries a negative charge. Note how positively charged species stick to this negatively charged bead. A cation exchange column holds on to cations and allows anions to pass through the column more quickly. Note that the structures of these resin beads all have a negative charge in order to attract and bind the positively charged cations. And I'm giving you some examples of popular cation exchange media, including Dow X50, a weakly acidic carboxymethylcellulose, and a weakly acidic chelating polystyrene resin known as Chelix 190. An anion exchange column holds on to anions and allows cations to pass through the column more quickly. Notice that the resins used in anion exchange chromatography carry positive charges. These positive charges will attract and bind the negatively charged anions. Here's an example of an ion exchange column. In this example, the resin used carries a negative charge. Thus, it's an example of a cation exchange column. The mixture of amino acids is poured through the column, and the amino acids will move through the column at rates determined by their net charges. Remember, the pH of the solution being used will determine the net charge of the amino acid. Since the beads are negatively charged, they repel amino acids with the strongest negative charge, but will hold on to amino acids with the strongest positive charge. In terms of the order of elution, anions with the biggest negative charge would elute first from the column, and we would expect these amino acids to be found in the earliest fractions collected. Anions with smaller negative charges would be collected in the next fractions. Cations with smaller positive charges would elute next, and finally, cations with very large positive charges would be in the last fractions collected. So the steps of ion exchange chromatography begin with selecting the appropriate ion exchange column. In the example we're going to work through, we're going to be using a cation exchange column, one with negatively charged beads. So then the next step is to prepare the amino acid solution in an acid such that the pH of the solution is below the lowest possible pK. This ensures that all of the functional groups of all the amino acids will be protonated and therefore will stick to the negatively charged beads of the column. Once the amino acids have stuck to the column, we want to be able to knock the amino acids off the column, but in an orderly fashion. We have two options here. We can increase the salt concentration of the buffer solution running through the column, or we can increase the pH of the buffer solution running through the column. Increasing the salt concentration works because the cation of the salt will compete with the positive charge of the amino acids for the negatively charged beads. The amino acids with the strongest positive charge will remain bound to the column, while those with weaker positive charges will fall off the column more easily. Increasing the pH of the buffer running through the column will begin to deprotonate the functional groups of the amino acids. This makes the amino acids less positively charged and more likely to fall off the column. On the right side of this slide is a very handy table that gives the pKa and pi values for every amino acid. Using these data, you can predict how amino acids will behave under different conditions. So what order will the following amino acids be eluted from a cation exchange column? The amino acids in your mixture are aspartate, serine, and lysine. The buffer pHs used to elute the amino acids are at pH 3, 7, and 10.5. The answer is that aspartate will elute first, followed by serine, and then lysine will be the last to elute. As an ion exchange column runs, fractions of a set volume are collected. The composition of these fractions reflect the binding characteristics of amino acids to the cation exchange column. These fractions can then be analyzed to identify the individual components of the original mixture. This analysis can generate something called an elution profile. At the top of the elution profile, you can see how the pH, and in some cases salt concentrations, of the buffers that were running through the columns change as the ion exchange column is run. At the bottom is the volume of the effluent that relates to the fraction number. We can then test each fraction for its absorbance in order to determine what the elution profile for that particular sample is. Once again, aspartate, which has an acidic side chain, is the first to emerge, where arginine, which has a basic side chain, is the last to emerge. Now, so far we've focused very much on separating amino acids from mixtures, but it is very common for these types of assays to be used for protein purification. So when testing chromatographic fractions, it's important, first of all, to make sure that the buffers you're using to elute your samples won't interfere with any downstream analyses that you're going to carry out. Typical assays that are carried out during protein purification, first of all, include the Lowry or Bradford assays. These are useful in determining the total protein content. We're going to talk about protein content assays in another lab. Sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE, can be used to estimate the purity of the sample. A nice crisp band tells you that you have a single protein, but a smear would indicate either protein degradation or impurities. You can also assay your proteins using immunospecific interactions with antibodies by Western blot analysis. And if you're testing for activity, ELISAs are another immunospecific assay that can be useful. Finally, if your protein is being expressed with a specific tag, you can use a number of techniques to detect those tags.